Greetings to all. Thank you to the conference organizers. I am Yerly Useche. I'm PhD in Biology and Research Fellow at the National School of Public Health, Sergio Aroca, at the National Institute of Infectology Evandro Chagas, INE, belonging to the Oswaldo Cruz Foundation in Rio de Janeiro, Brazil. Today, I'm going to present you the theme, Purinergic system involvement in cardiovascular diseases, following the next aspects. First, the relationship between the purinergic system and inflammation. Second, how hyperinflammation can cause cardiovascular diseases. Third, which are some of the regulation mechanisms of the purinergic systems over main cardiovascular diseases. The purinergic system is composed by extracellular nucleotides and nucleosides, the receptors, and the ectoenzymes that are what are responsible for their levels regulation. The ATP is mainly released for damaged, stressed, or infected cells. However, in physiological conditions, ATP is also released from cells with a high ATP intracellular concentration and from vesicles containing neurotransmitters and hormones. Extracellular ATP is transformed in ADP and later in MEP through the membrane ectonucleotidase CD39. Then another ectonucleotidase CD35 converts ADP into adenosine. Each one molecule is recognized by several receptors expressed in membranes of diverse kinds of cells. ATP activates cation canals that exchange calcium and sodium cations for potassium cations. These are named P2X1 to 7. Moreover, ATP activates G protein coupled receptors called P2Y. Some of these receptors are also activated by ADP and uridine-derived nucleotides. Adenosine activates G-coupled receptors by the type P1A123. Therefore, the functions accomplished by the purinergic system depends on the concentration of nucleotides or nucleosides and the kind of receptor and ectonucleotidases expressed on membrane of diverse cells. But what is the relationship between cardiovascular diseases and endothelial dysfunction? The oxidative stress has been described as a major factor for the development of vascular diseases. The stressor agents include excess of macronutrients leading to hyperglycemia or hyperlipidemia, infection by pathogens, contaminant elements, and hypertension. Elevated oxidative stress leads to endothelial dysfunction, characterized by impaired vasodilation and deficiency of nitric oxide, and activation of endothelium, that is an inflammatory state, which are the base of several cardiovascular diseases, including hypertension, angiogenic diseases, atherosclerosis, calcific aortic valve disease, thrombosis, stroke, and fibrosis. Moreover, different roles of the purinergic system have been revealed intervening in these cardiovascular diseases. For instance, in hypertension, different combination of agonists, receptors, and cells induce vasoconstriction or vasodilation. Also, purines promotes migration of endothelial cells and proliferation of both smooth muscle and endothelial cells during angiogenesis and atherosclerosis. What's the role of the purinergic system in hypertension? The development of hypertension is consequence of several mechanisms, including increased vascular resistance, largely determined by the increase of vascular contraction and arterial remodeling. Moreover, the purinergic system influences in this vascular contraction-dilation equilibrium. The vasoconstriction is consequence of the interaction of several combination agonist receptors. For instance, ATP released as a co-transmitter from sympathetic nerve links with the receptor P2-CYP1 or 2 led to the entrance of calcium-inducing contraction. But the other way, some P2A and P1A receptor associated to G protein are expressed in the smooth muscle and activated with ATP or adenosine-inducing vasoconstriction. For example, ATP released from sensory motor nerves in interaction with PY1 or 6. Moreover, this ATP is transformed in adenosine, what activates the receptors P1A1 or 3, leading to vasoconstriction too. Another agonist receptor pair that induces vasoconstriction is the UP4A, uridine adenosine tetraphosphate, which is released by the stressed endothelium and activating the receptor ionic P2X1 and protein G-associated P2A2 or 4 in the smooth muscle. Conversely, ATP released from endothelial cells, erythrocytes, or platelets, mediating vasodilation via endothelium-derived nitric oxide through activation of P2Y2 or 2 and P2X4 receptors. Additionally, adenosine activates the receptor P1A2, leading to vasodilation too. Moreover, as we have seen, the inflammation is a factor to increase hypertension. Inflammation increases the reactive oxygen species released by the activated immune cells, promoting the endothelium dysfunction and several vascular cells death. For instance, circulating ATP can activate the ionic receptor P2X7 in monocytes, leading to cytokines production and hyperinflammation. What's the role of the purinergic system in atherosclerosis? First, let me point out the process of atherosclerosis development because purinergic system regulates some of them. As we have seen, several stressors cause activation of the endothelium. That's the beginning of the atherosclerosis and all related diseases. First, upon activation, endothelial cells express adhesion molecules that promote the recruitment of immune cells, such as monocytes. These cells then infiltrate the arterial intima, where monocytes differentiate into macrophages and interact with neutrophils and T cells, while endothelium is repaired by proliferation and migration. Second, increased entrance of lipoproteins LDL, cholesterol, into the intima and later oxidized LDL are uptaken by macrophages, promoting foam cell formation. Macrophage foam cells and other cells can aggregate, die, and disintegrate, thereby forming a plaque or necrotic core. The smooth cells migration and angiogenesis of vasa vasorum promotes plaque progression and remodeling. Fourth, the endothelium covers the plaques and calcium salts are accumulated in the plaques. After, blood clots are formed over damaged endothelium. Those can rupture due to intraplaque hemorrhages, forming an arterial thrombus which can lead to a myocardial infarction and a stroke. But how is the purinergic regulation of atherosclerosis? When endothelial cells are activated, two sets of processes are regulated by the purinergic system. First, proliferation and migration of specific cells. Adenosine activates the receptors P2A or 3 to induce proliferation of endothelial cells, as also ATP and UTP through receptors PUA1, 2, or 4. Moreover, the last set of interactions induce vascular smooth cells proliferations and dendritic cells activation. The second process regulated by the purinergic system is the leukocyte recruitment and their inflammatory response. For instance, monocyte recruitment is induced by the activation of the receptor P2A2 by UTP, while adenosine counteracts this action, activating the receptor P2A2. Leukocyte recruitment is induced by ATP in interaction with some P2A and P2X receptors, while the administration of ectonucleotidase CD39 restricts this action. Adenosine also regulates neutrophils depending on its concentration. Low adenosine concentration leads to chemotaxis and phagocytosis by neutrophils through PA1 or 3 receptors, while high adenosine concentration leads to inhibition of inflammatory response through PA2 receptors. Additionally, endothelial cells increase their inflammatory response by UTP or UDP binding with P2A6 receptor. Conversely, adenosine also could induce protective response by macrophages, producing anti-angiogenic factor SFLG1 through PA2V activation and reducing cytokine production through PA2A. In the later phase of the atherosclerosis development, the adenosine is also atheroprotective. In interaction with its receptor PA2A, inhibits the formation of some macrophages, and the administration of the ectonucleotidase CD39 decreases the neointimal hyperplasia. Another critical process of are the clot formation and the calcification of the plaque. The platelet aggregation is induced by the ADP binding to P2Y1 or 12 receptors and by the adenosine interaction with P2A2 or 3 receptors. Moreover, ATP can activate the P2X7 receptor in monocytes, inducing cytokine production, inflammation, and prothrombotic statement. For instance, clopidogrel, ticlopidine, clagrelol, and others. Also, CD administration decreases platelet deposition, while antagonists of P2X7, as given in Clamid, decreases the inflammation. By the other way, the calcification of the plaque is induced by the UP4A activating P2A receptors, while ITP binding P2Y2 receptor is protective against the mineralization of the aortic valve. Also, inhibitors of ectonucleotidase CD39 prevent mineralization too. As conclusion, the regulation of cardiovascular diseases by the purinergic system is highly complex. Maybe for this reason, the therapeutic administration is a challenge. Approximation of nutrition-based treatments lead to decrease oxidative stress have been recently abroad in the hope of cause less adverse effects. Thank you for your attention. These are the reference used for this presentation. Thank you.